Good afternoon, I'm Nicole Wynn here on Science Mania. Today we're going to be talking about cloning. Is cloning the right thing to do, and should cloning be allowed? But first, to our science expert, Maggie Brock, for more details about the cloning process.
Thank you, Nicole. I'm Maggie Brock, here to explain how cloning works. There are many different ways to clone an organism, but today we're going to close in on the process of nuclear transfer. In a science lab, a scientist obtains a cell from an organism. In this case, the organism shown is a frog. For explaining purposes, the skin cell will come from frog A. The scientist then extracts the nucleus from the skin cell. The egg cell then obtains from frog B. Using radiation, the nucleus of the cell is destroyed. The scientist then implants the nucleus in the skin cell of frog A in the egg cell of frog B. The egg then continues to develop and turns into a tadpole. This tadpole is a clone of frog A because it contains the DNA of frog A. Back to you, Nicole.
Knowing the process of cloning, it's time to inform our viewers of the pros and cons of cloning. Here to tell us about the positives of cloning is Kaylee Holmes.
Question around the world, but there are many useful things that can come from cloning. Cloning is a process that can be used to study and research diseases. By doing this, we can potentially find cures for diseases. Researchers at Oregon Health and Science University in Beaverton cloned monkey stem cells. These stem cells are very useful because they can be developed into any tissue or organ. Also, Dr. M's next project is to use cloning to create monkeys that carry genes for human diseases. He uses and studies these monkeys to understand the cause and treatment of the disease. In addition to using cloning to study diseases, cloning can be used to revive endangered species. In January 2001, scientists delivered a cloned baby gaur. These scientists used a cow to produce a baby gaur named Noah in 2003. Advanced cell technologies produced the first bandage and endangered species cloned from adult cells. Cloning can be used for transplants. In February 2002, scientists from the biotech company Advanced Cell Technologies created a clone cow embryo. They let it develop into fetuses and harvest the fetal tissue. They transplanted the tissue into the donor cow. No signs of immune rejection to the transplant were shown after 3 months of observing the cow. Also, scientists at the University of Missouri created 4 cloned piglets. One copy of a sugar-producing gene in the pigs was knocked out. The sugar-producing gene is rejected when organs are transplanted into the humans. Since the gene is knocked out, the transplant has a better chance of being successful.
The positive side of cloning sounds reassuring, but in the science world, there are always complications and errors. Here today to tell us about the opposing side of cloning is Susan Brown.
Like any other issue, cloning also has its downsides. Although cloning can be used to benefit the world, it's necessary to know the effects of cloning. The biggest issue with cloning is the moral question. Where should we draw the line? Can an organism be cloned? Director of Education at the National Catholic Bioethics Center questions if it will be a moral justification to clone human beings. If cloning has to be a moral question, can it really be considered right? The Humane Society of the United States encouraged pet cloning because many animals in shelter are desperate for homes. People should not be allowed to clone something where there are plenty of animals that need homes. A moral question is not the only thing in play when talking about the downsides of cloning, though. Cloning is also in danger to organisms. If the process of cloning is successful, then it still causes premature death. Scientists use a cow to produce a baby gaur named Noah. The gaur died 2 days after birth. It's not right for a scientist to bring something to life and then let it die 2 days later. It's also, according to the Center genetics in society, at least 95% of cloning experiments fail in the form of miscarriages, stillbirths, and life-threatening glitches. If a cloning organism doesn't die a few days, the organism has a chance of functioning abnormally. In 2002, a study of the Whitehead Institute of Biomedical Research on Cambridge, Massachusetts, on mice indicated that 4% of genes function abnormally. In addition to this, Australia's first cloned sheep acted completely normal on the day it died. The research of its death is still unknown. Cloning should not be allowed because many animals cannot function properly. It's a question whether or not cloning should be allowed. The cloning is a dangerous process with many negative outcomes. The use of cloning should not be allowed in research. Back to you, Nicole.
As new developments continue to occur in the science world, it's difficult to decide whether a new development is a positive or negative change to our world. Cloning is an issue where there are many rights and wrongs. With cloning, scientists can research and study diseases for new cures. It can also be used to revive endangered animals. Cloning can also be a future solution for transplants. On the other hand, cloning can cause organisms to function abnormally and cause premature death. A moral question also comes into play when talking about cloning. With the issue of cloning, it is a matter of opinion. With so many pros and cons, it's hard to decipher whether or not cloning is right. Thank you for watching our segment on cloning. See you next time on Science Mania.